Today we're going to look at the science section of the ACT and look at some tips and a little bit of practice on how to do well with the science section. It's the last section of the ACT, which is kind of a big deal because, as you probably know, the ACT takes a while. When we do it here at school, we start at 8 and we essentially end at 11:30. So, I mean, you're looking at really, that's nearly 3.5 hours of testing. I mean, that's a long time to be working. And so when you get to the ACT science section, you've nearly tested for 3 hours at this point, and you're going to be pretty tired. So fatigue is a big deal when it comes to the ACT, the science section of the ACT. But it is the last section. It consists of 40 multiple choice questions, and it takes about, you have 35 minutes to take it. One of the interesting things about the science portion is it is not content-based. Technically speaking, I guess you do need to know some science terms, which we'll talk about a couple of those like variables. But realistically, you should be able to not know anything about chemistry and get all the questions right on a chemistry section of the, a passage about chemistry. So you do not have to know the specific content. Now, sometimes that can help. Like I remember seeing a section where it talked about momentum, and it was nice to kind of know what momentum was, but you do not have to know these things in order to do well. There are 3 different types of passages. The first one is data representation. And so in data representation, they're going to have like graphs, tables, and charts. And you have to identify, different data points. You might even have to infer some data. So you might say, it might tell you what the temperature was at 3 p.m. and what the temperature was at 7 p.m. and then ask, okay, what would the temperature be at 5? And you sort of have to look between the lines there and say, well, it should be somewhere in between those. And try to pick out the best. So a lot of times they'll ask you to not only look at, okay, what was the temperature at 3 p.m., but then you have to infer a little bit more at times. The biggest tip, and I'm going to show you one of these graphs in a minute, but the biggest tip is really to look at the labels, and that's a huge deal, the axes labels, X and Y. See what is being graphed. Another thing is that a lot of times there are, and this goes for really the first and second parts, there are these like passages, some text there that kind of, you really only need to read once because most of the information is going to come from outside the text. Most of the answers that you need are going to come from the graphs and tables, but on occasion there is something that is in the passage. So for example, this I pulled from a practice ACT. So if you have these 2 graphs, I mean, how do you start analyzing this? And again, to me, the biggest thing is you need to look at the axes. And I know you'll forget, so let me show you again. This is the y-axis that goes up and down. So this would be my y-axis. And I remember that because if I think about the capital letter Y, I go up and down a lot when I write it. Okay, so up and down is y-axis. So here we see on the y-axis we have, it looks like these are days, the 11th day, 12th day, and so forth. And on the x-axis, which of course goes the other way, on the x-axis you have, it looks like proportion of eggs hatched. So we're talking about hatching eggs, which makes sense because the title is Hatch Time, Trial 1. So again, it's very important, and we have a legend over here which says 380, and I don't know what that is, ATM. Now, I actually do know, I think this is micro atmospheres, but even if you don't know, it's not a big deal. It's just, okay, whatever that is, 380 ATM, that's this one, and this is the red one. And so again, it might ask you to compare trial 1 and trial 2, but if you know what the axes are, that would really help you. The second type is called research summary. They usually ask you to compare 2 or 3 different experiments and tell me, what's different, which one is might confirm more information, that sort of thing. My tip here is to, in these experiments, identify your controls and variables. And those are really important science terms that a lot of times I think we forget about. So let me tell you what a control and variables are. So first of all, the goal of an experiment, anytime you do it, is to see how one thing affects another. That is ultimately what you want to see. And specifically, instead of saying one thing affects another, what we're really saying is how does the independent variable affect the dependent variable? And it might seem sort of silly. Like, what if I said, how does the color of bulletin board paper, we'll just say paper, but How does the color of my bulletin board paper affect student test scores? Now you might say, well, Mr. King, that's stupid. Of course they don't affect it. It doesn't matter what color the paper is. But scientifically speaking, you don't know that until you do an experiment to see. Maybe it does. I mean, if you haven't done it, you don't know for sure. That's the essence of doing science. And so in this scenario, these two would be my variables. Now the control is what things have to stay the same. Well, think about it like this. If I gave one group of students a test that was about algebra, basic calculations and operations like addition, subtraction, multiplication, but then I gave another group a test that was about calculus, I can't really say that that really the color of paper affected the scores because I didn't control all the variables. So in that experiment, I'm trying to see how does my bulletin board paper affect student test scores. I need to make sure that I have roughly the same types of students, that I have the same test. There are a multitude of other things. I have to make sure that I'm not playing extra music during one class while they're taking a test, or that if I'm going to be up around, I need to be moving constantly throughout. So I have to keep everything the same except the bulletin board paper, so that I know for sure that that's really what's affecting the student scores. So controls are things that you have to remain the same. That might be, if you're doing a classic experiment, it's like how does fertilizer affect plant growth? Well, you need to make sure you have the same type of plants. You need to make sure that they get the same amount of water. You need to make sure they get the same amount of sunlight. So there's lots of variables there that you have to keep the same, and that's the control. Now, independent, dependent, how do I know which one is which? Well, independent variable is what I can control. And so the independent variable between these two, color, paper, student test scores, I can't control how well they do. I can control the color of the paper, though. I can make it red, I can make it green, I can remove the paper, I can do those sort of things. But that's the independent variable. And then here the dependent variable would be, the scores themselves. That's what I measure. And you can even extrapolate this to a graph. If I was going to make a graph, you usually have an independent variable on the x-axis and the dependent variable on the y-axis. So again, I could even go ahead and graph this. I could have my coloring paper here, so maybe I have like red and blue and green and yellow, and then I would do maybe like a chart, like, when red, it was here, black, it was here. Anyways, you get the idea. So maybe I found out that yellow does better. And again, that's not the case, but I haven't tried it, so I really don't know. But anyways, so if you can identify those parts of the experiments, then it's going to be much easier to compare. Be like, this is experiment 2 wasn't very valid because they didn't keep everything the same. They didn't take care of their controls. The last one I want to talk to you is probably the hardest one, which is called conflicting viewpoints, or I like to call it dueling scientists. So you're going to have two different theories, and you have to, they're going to give you two different theories from two different scientists, and you have to compare them. And that may even sound like ridiculous theories, if it's something, it may sound silly, but you still have to, and I think that might be what makes it hard, you still have to be able to be like, this theory is valid in some ways because of this, or this piece of information refutes that theory, even though I kind of like that theory, or whatever it is. So you kind of have to detach yourself from what you like or don't like or think is reasonable or not, and actually just look at the theories themselves. And so one of the tips is really what I would do is, again, you can write on your test. I see all the time people don't really write very much on their ACT, but you can write as much as you want, basically. So what I would do is I would really summarize the differences while you're reading. Okay, so what you have before you is there should be a passage. It should have questions 37 through 41. I want you to read the passage, pause the video, and answer the questions, and then when we come back, we will go over the answers. So go ahead and do that now. All right, welcome back. Hopefully you didn't think it was too hard, but let's go over the answers and why they're correct. So let's start with number 37. It says, according to theory 1, the compound CH2Cl2, and then it wants you to identify which of these is the correct answer. So I know from theory 1, it says that it talks about how there has to be this 8 electrons surrounding the carbon. Well, I can see that there's not here, right? There's only these hydrogen, right? And so I know that there's not, so 37, the answer is B. It cannot exist because the original carbon atom does not have 8 electrons surrounding it. Now this is the one that when I did the practice I actually missed. I thought the answer was A. It should have 8 electrons, but it doesn't, right? So I think what they're trying to say is that this compound does have 8 electrons surrounding the carbon atom, and it doesn't because there's H2 there. So that one was the one I missed, and hopefully you got it right. 38, according to theory 1, the compound xenon F4, I don't know, whatever that is, there's this compound. Now that was actually the one they talked about. And so this one, they talked about how it refuted theory 1 basically. So according to theory 1, this shouldn't exist, and that's what it says straight up from the passage, which you can look up. And that's the correct answer, that it should not exist because xenon already, it said already had 8, but it shouldn't have any more. So then this couldn't exist, but then it did, and so that's why theory 2 came into popularity. 39, the atom boron has 3 outer electrons in bonding to boron. A fluorine atom donates 1 electron. The BF3 molecule is known to exist. Which of the following is true? So again, a lot of times there's this jargon and we get confused about it, but they showed you that picture where they were all kind of spread out. Well, if you know that BF3 has 3 valence electrons, what's the best way to spread those out? First of all, it doesn't follow theory 1 because, well, there's only that one right there, but I don't know how it has 3, so 3 and 3 is 6, and there's supposed to be 8, so it doesn't work. It doesn't necessarily contradict 2 because there's some pairs there. What happens is that you know it's going to be one of these 2, so then which of these makes more sense? And it talks about how they want the electrons to be evenly distributed, so it's got to be D, which is the correct answer. 40. A scientist seeking to explain why theory 2 has more predictive power than theory 1 might argue that 8 electrons shall represent a closed shell. While 8 electrons represent a closed shell for some atoms, for others the closed shell may be 6, 10, or 12. It is incorrect to assume that a given atom always has the same number of electrons around it. CH4 is not as important a compound as XeF4. So here, what really happens is, because this is theory, theory 2 is better because what was the problem with theory 1? Well, theory 1 was all like, oh, it's got to be 8, it's got to be 8. And then they're like, but wait, this thing has not 8. And so this one, theory 2, has more power because it predicted that there's some where, again, they might have 6, 10, or 12 sort of atoms or electrons in the closed shell. So that's why F is not correct there. And finally, 41. Theory 2 can be threatened by evidence of. And so theory 2, we remember, it's basically talking about how there's all these pairs, right? Well, if we have, you'll notice that this kind of is like an even number, so there probably are pairs there. This has an even number, so there are pairs there. Molecules with stable octets are fine because there's pairs there. So which of these is the only one without pairs? Well, you have this F5. How's that going to work? And this is the correct answer. So if they found XeF5, then theory 2 would be out the window because it talked about all these pairs, and all of a sudden we have 8 electrons there, and then presumably 5 electrons from the fluorine there. So again, these type of passages are the hardest ones, and really what it comes down to is you need to identify what was different. You know, the first one was all about 8. Theory 1 was like, oh, there has to be 8 electrons, whereas theory 2 was like, hey, you know what, we can do these that have 6 or maybe have up to 12. And that was the big difference, and that's kind of what guided me through these questions. So hopefully you found that helpful. Thanks for watching.